So far we've seen the big picture, the big folded up proteins, and then we saw the details, the individual amino acids. And so now we're going to combine these amino acids and see how they end up looking like something like hemoglobin. So we're going to dive into how a protein folds up, and protein folding is an area that a ton of research is done on because it's really useful to us in a lot of different ways. So we're going to start with a couple of big pictures and then get into some more details here. So when we talk about levels of protein structure, we'll see the words on the next page also, but we're going to talk about primary structure, secondary structure, tertiary structure, and quaternary structure. Same words we've used to describe alcohols and some other things before. And we'll look at more details in a second. But primary structure is going to be the fact that we just string those amino acids together, right? And we always have what functional group holding them together? Amides or peptides, right? And here are those amides or peptides in the primary structure holding amino acids together. So saying here's one amino acid connected to another amino acid connected to another amino acid, and remember those connections are what I have highlighted in orange here. So we can connect our amino acids, but then they start to fold up and have intermolecular forces between them. And so I'm going to leave down here a little note to you that we can have intermolecular forces holding these together, and the color I'm going to use today for intermolecular forces is going to be purple. And you see a bunch on here. Anywhere I have these dashed lines, you want to be thinking about these being intermolecular forces. So this would be my long chain of amino acids, and suddenly these attractions show up. Even where there's no dashed lines, there's still attractions there, holding this protein into the cool ribbon shapes that we can see here and that we saw with the hemoglobin earlier. So I just colored in a few, but there's a bunch of those weak attractions between molecules, and that's what we're going to dive into more on the next page. There's also one special type of attraction we'll see in a minute where these two sulfurs actually have a covalent bond between them. And so covalent bonds, if you can remember, are a lot stronger than intermolecular forces. So this is a super strong link between those amino acids. We have some of those in our hair, and if you ever perm your hair, you use chemicals to break those strong bonds and reform them into the curls or the straight hair that you would rather have. So we are going to look at these different levels of structure, and we'll come back to this page to see where they fit in here, but we're looking at these attractions that make this straight amino acid chain kind of fold up into a cool, more complex shape. So as we move on here to the next page, let's define in a little bit more detail our primary, secondary, tertiary, and quaternary structure. So our levels of protein structure, primary we're going to look at first, obviously, and this is going to be the sequence of amino acids joined by peptide bonds. And so one way that you might see this written, you might see it written alanine is bonded to glycine, is bonded to valine, and it keeps going there, all right? These peptide bonds, remember, are amide bonds. These lines here would be my peptide bonds, and that's what we just saw on the last page, these carbonyls next to an NH. Those are your peptide bonds that are holding this whole polymer together. So that's primary structure. It's just a straight, boring chain. Then it starts to fold up. So secondary structure is the 3D arrangement of local regions, so not the whole entire thing, just small regions of the protein. And what we'll see here is we'll see hydrogen bonding. So back to that chapter 4 concept. Hydrogen bonding is going to happen in the backbone, and it's going to happen between the NH that we see in our peptide bond and the carbonyl. And so again, this is going to be one of those purple attractions that we colored in on the last page. And there's 3 different types of secondary structure. We actually saw them on the first page. Alpha helices, beta pleated sheets or beta sheets, and then unstructured regions. And so if we can go back here to the previous page, what I see here with this helix, this is an alpha helix here that I'm looking at. And then over here where I have these parallel sheets, this would be a good example of a beta sheet. And you can see here these attractions that are holding the spiral in a helix shape. Those are the attractions between the NH and the C double bond O. And here's actually one in a beta sheet. It's in the backbone, so here's that NH hydrogen bonding to the CO. Remember, for a hydrogen bond, you needed a donor and you needed an acceptor. So there's your donor and your acceptor, and they're hydrogen bonding. And those are within the backbone. These are not in the backbone, these other ones that kind of stick out, and we'll see those in our tertiary structure. All right, so alpha helices, beta sheets, that is secondary structure. And here's a close-up also of the alpha helix and some of those attractions. You can see again the carbonyl and the NH. It's all very small, but there would be your attraction that kind of holds these helices into that spiral shape. And there's more of those attractions throughout this alpha helix. So here's my secondary structure that we just looked at, sheets or helices. Third is tertiary structure. And here you can see there's a bigger space, so there's more to say about it. This is going to be my overall 3D shape adopted by one polypeptide chain. So adopted by one polypeptide chain, one strand, all right? And this is going to be due to side chain attractions. So notice that my secondary structure was backbone interactions. It was those amides interacting. The tertiary structure is going to be side chains interacting. And if we go back over here to this page, we can see some of those different attractions. So something like this disulfide bond, those are side chains. Those are cysteine side chains that are interacting with each other. Or here is a basic side chain interacting with an acidic side chain. And so those side chain interactions that are outside of the backbone and just reaching into that in-between space, that's what's going to hold together my tertiary structure here. So there's 4 different types of side chain attractions that we can see for tertiary structure. I'm going to label them A through D. So the first and the strongest is called a disulfide bond, or sulfur bridge, sometimes you'll hear. So disulfide bond, strongest ones. Next will be electrostatic attractions. Also very strong. You could go back to chapter 4. There was a table where there's space for those to be added. Polar attractions, we've seen those before. And then the weakest will be my nonpolar attractions. Those nonpolar attractions, another word for them would just be those dispersion forces that we've seen before. Polar attractions, we'll see there's actually 3 different options for those. There are ion dipole, hydrogen bonding, and dipole-dipole. So 3 different types of polar attractions. So those are going to be those side chains reaching across the gaps to each other. Finally, looking at quaternary structure, that's my last one here. This is what we see all the way back at the beginning on the first slide when we saw hemoglobin, which I've lost track of for a moment. Here it is. In hemoglobin, we saw that we had 4 polypeptide chains, and so we need something to hold the 4 different groups together, the 4 different subunits, and that's going to be these attractions that hold quaternary structure together. And so quaternary structure is going to be the overall 3D shape adopted by at least 2 polypeptides. So you have to have at least 2 chains or 2 subunits. And these again are going to be due to your side chain attractions. And these are going to be the same attractions as tertiary attractions, but rarely do we see the disulfide bonds. So it's going to be mainly B through D that we saw up here. So between 2 chains, you'll see more of these. If we can go back to this page here, all of this is one polypeptide chain. And so in order for us to have a quaternary interaction, I would have to have a separate unconnected polypeptide chain nearby. And if I have attractions here between one polypeptide chain and another, then this would be my quaternary attraction, two unconnected polypeptides that are attracted. So those are our definitions of our levels of protein structure. Next up, we will look at them in some more detail.